For this part of the lab, we are going to compare the effects of ethanol versus water on the pigment in the cells of the onion. In preparation, I have filled two 6-dram vials, this one with 10 milliliters of ethanol, 40% ethanol, this one with 10 milliliters of deionized water. I've taken an onion and I've cut that onion into small pieces. So this is our original onion piece. It has plenty of pigment. Then I cut it up into small pieces that have both white and purple parts in them. I tried to make the pieces pretty much the same size, but you can see that there's slightly different sizes in there. I'm now going to weigh out approximately 6 grams of onion tissue, place it into each of these two solutions. So we're going to now look for approximately equal size pieces. Make sure I'm not biased on the color of the pieces as I grab them and put them on here. So I need to make sure that I don't bias the experiment in any way. But these pieces are very different in how much pigment they have relative to no pigment. And their sizes and shapes also are variable. So that's something to pay attention to, is the variance in the sizes and the shapes and the amount of pigment in each of these. Need a really small one then to make sure. Getting exactly 6 is pretty tough. Close enough. We'll go 6.02. I'm going to take all of these then. We'll place them into the vial with the ethanol. We'll go here, tear it, and get 6 more to be placed in the other vial. Last piece just needs to be really small. 6.01 versus 6.02, close enough. So we'll go ahead and put these in this vial. Now you'll notice that the solution in both of those is down at the bottom, so I need to take this, push it down, make sure that all of those tissues are below the surface of the solution. Take this, push it down, make sure that everything is below the surface of the solution so that we have them ready to go. And in 30 minutes then we're going to take those out and do the next step. The next step is to take each solution and estimate the amount of pigmentation in the solutions. To do this, we'll use a spectrometer. The spectrometer hooks up to the computer and runs on software, and basically is going to be used to measure the amount of light that gets absorbed by the pigment in the solution. So it's basically going to give us an estimate of the amount of pigmentation in the solution. To use a spectrometer, then I need these little features called cuvettes. You'll notice that the cuvette has a side that is clear and a side that has all these ridges in it. The clear side basically goes into the spectrometer where light shines through. The spectrometer knows how much light it is shining through. It knows how much light gets to the other side, and it then uses that information to estimate absorbance at whatever wavelength that we want it to use. Over here then I want to basically take my solutions and put them into the cuvettes. So this cuvette is labeled, it's experimental for DI water. Here's our DI water. I'll take this then, mix it all up, make sure it's ready to go. We will take our pipetter and put a lid on. Our pipetter then is set for 1,000 microliters, which is 1 milliliter. I will then go in gently, suck up 1 milliliter, put in 1, go in, gently suck up a 2nd milliliter, and we will then actually get a 3rd milliliter. So we have 3 milliliters in that solution. I'll change pipette tips. We then are going to put this into the experimental ethanol and get this out. We'll stir it around like we did the other one. Then we shall go in and get 1. Okay, a little bit of drill here. 2, and our 3rd milliliter. 3 milliliters of that. And now I'm going to go hook the spectrometer to the computer and we will do the analysis. Now that we have our solutions in our cuvettes, we can take the cuvettes and use a spec to measure the absorbance. So here's our spectrometer. It's hooked up to the computer with this cord. In front of the spectrometer now we have 4 different solutions. We have the experimental DI solution, which is onion in DI water that we just made up. We have the experimental ethanol solution, which is onions in the ethanol that we just made up. I'm adding 2 new solutions now. One is called BDI or blank DI, which is just straight DI water. The other is called BETOH or blank ethanol. I'm going to start by taking the blank water, putting it into the spectrometer to calibrate the spectrometer. Okay, the next step is to determine the absorbance of the solutions. So here we have an image on the screen. This is Logger Pro, which is the software that works with our spectrometer. We can see up here our y-axis is absorbance, our x-axis is wavelength. Wavelength of light determines the color of light. Down here we have about 380 nanometers, which is purple, a little over 400 is blue, then a little over 500 turns green, then to yellow, then orange, and up here around 700 nanometers it's red light. So that's all visible light, and we're looking at visible light when we look at pigments because pigments are what we see within that visible light. Now when we think about absorbance, there are three different types of light. There's absorbance, there's reflectance, and there's transmittance. Transmittance is when light that basically passes through an object. So when you have a see-through object, and that see-through object may be colored, the color of that light is due to the wavelengths of light that are passing through the object to your eye. When you have a solid object, then you're looking at reflected light. Reflected light then is light coming in. The wavelengths that bounce back to your eyes most are the wavelengths that you see, which determines the color. So something that is green right here, this is due to reflected light bouncing off back toward your eye. Finally then, absorbance is the light that doesn't bounce back. Okay, so if you think about those things, then perhaps, right, the absorbance, then if something is purple in color, you'd expect it to be reflecting, and you'd expect it maybe to have a high absorbance. Okay, things are more complex than that, and we'll show you that in a minute, but that's a good start for the color of light. Now what we're going to do here is we're going to use the spectrometer to measure the absorbance of the light in the solutions from the onion. So to start with, what I'm going to do is I'm going to take straight water. This is our blank DI water. Place it in the spectrometer. Go up here to experiment, go down to calibrate, and calibrate the spectrometer. Now I already warmed it up, so I can skip the warm up and finish the calibration. Okay, it only takes a couple seconds to calibrate. Then I'm going to hit OK down here. All right. Now what calibration is that calibration is basically tearing the scale. So if I hit play up here, what we're doing then is we're setting the amount of light that gets through all of the wavelengths of light in water to 0, right? So water has its own absorbance qualities, and we're basically tearing all of that to 0. Now what I'm going to do is I'm going to take my blank solution of water, take it out, put it to the side, and now find my experimental DI water. This is onions soaked in DI water. I'm going to press play up here. I'm actually going to erase and continue, get rid of that red line, and then I'm going to press stop. And this basically is where we're absorbing. So onions are purple, which is interesting because here we're actually reflecting off all of this blue light clear up through the red. We're actually absorbing purple. So the fact that our onions are purple means that we're actually reflecting off this purple while we're absorbing this purple, and then that changes the color of purple that we actually see reflecting off from those onions. Now what we want to do is I want to get an estimate of the amount of pigment in the solution. We're going to get that estimate by the height of this absorbance peak. 390 nanometers is right about there. Okay. And so we're going to use that absorbance at that wavelength to determine how much pigment's in the solution. So if I scroll down here to 390 nanometers, 390 then our value is 0.169 optical density, or O period D period. So 0.169 O period D period is the value that you want to record. Okay, now what I'm going to do is I'm going to take this experimental DI out, switch it now with our blank solution for our blank ethanol. Put our blank ethanol in, go up here and hit experiment and calibrate. We're going to recalibrate it now with ethanol. Skip the warm-up, finish the calibration as we did before. Then I'm going to press OK. I'm going to take my blank out and put in my experimental ethanol, which is my onions and ethanol solution. Hit play up here, and we will store the latest run so we can compare both right on. And then I'm going to hit stop and finally go down to 390 nanometers, and the value is 0.976 O period D period. And that's the value to record in your lab.